All right, everyone, I want to welcome you to day 2 of our annual PKD symposium, sponsored by the PKD RRC. I think we have an interesting lineup today. The first of the morning sessions, we'll talk about some new tools that are being developed for PKD research. Then the second subject will be focused on energy utilization and the connection between the polysystems and metabolism. We have a training, we adjourn for lunch for 45 minutes, hopefully if we stay on time. The trainee workshop will follow after that, so please attend that and learn a little bit about up and coming junior scientists in the PKD field and how they made it there. And if you haven't registered for that workshop, you can still do that. So please email Stacy, the email's there, if you would like to participate in that. Next slide, Terry. So some just kind of housekeeping, the same system set up as yesterday. We'll have everybody muted and no cameras except for the speaker and the session chair and the panelists. We have a panel. We're going to try to handle all the questions through the chat, so please submit them so that everyone can see them. We may not have time to address all the questions during the meeting, and the speakers hopefully will address them as we go through the chat so that everybody can see it. For speakers, we'll give you a 3-minute warning when you come close to the end of your talk. Please try to stay on time. If you have any technical issues, please email John or Stacy through the chat. Sorry, not email, contact them through the chat. We'll try to resolve that as quickly as we can. And also as a reminder, after the meeting, we're going to send out an evaluation. This is really important to us as consortium to try to understand what we can do better, what you liked, what you didn't, and we will try to get back to you with answers. Next slide, Terry. So I want to thank again all those that put a lot of effort into organizing this meeting. The organizing committee is shown there, including 3 trainees who have helped to organize this meeting. And then, of course, the NIDDK for support of the symposium. And so with that, I'm going to turn it over to our first session chairs to introduce the speakers.
Yeah, thanks a lot, Brad, and also warm welcome from us. So it's Ryuji Morizani and myself who are sharing the first session, and we are well in time, I would say, so 7 minutes earlier. So enough room for discussion. And I would like to introduce the first speaker, who is Vishal Patel from UT Southwestern. He's an associate professor there and also the director of the PKD and kidney genetics clinics. His work focuses on RNA-based therapeutics to treat PKD, and he will present today PKD1 mRNA Cis-Inhibition and ADPKD. So Vishal, go ahead, the stage is yours.
Vishal, you may be muted. We're not hearing anything.
You're still muted.
You're still muted. Yeah. Vishal, you're muted.
Got it.
Ah.
Good morning, everybody. And thanks to the organizing committee for inviting me. It's been a great meeting so far, and I'm happy to kick off the second day. So instead of cool new tools, which is the title of the session, I will be presenting what I think or what we feel is cool new biology, that PKD1 mRNA is cis-inhibited in ADPKD. We think that this finding may provide the basis for developing a new class of PKD1 boosting drugs, which I hope you all will agree will be the ultimate cool new tools. So let me start by telling you the ins and outs about this, this new mechanism that we've discovered. So everyone here knows that heterozygous PKD1 mutations cause ADPKD. Loss of one copy reduces gene dosage by 50%, but this somehow is not sufficient to initiate ADPKD. Several lines of evidence now indicate that the dosage needs to fall even further by 70 to 80%, dipping below a critical threshold, which then leads to cyst initiation. This additional 20 to 30% loss can only occur if the remaining non-mutated allele is being repressed, considering that one allele has already been lost. So this brings me to my research question. Before that, I perhaps should say that this suggests that the remaining copy of PKD1 perhaps is just as important as the mutated copy in ADPKD initiation and progression. This brings me to my research question, which is, how is the remaining PKD1 copy regulated in the context of cyst growth in ADPKD? Any insights into this question may provide us ways to harness the remaining copy for what would be endogenous PKD1 gene therapy. So how do we get into this? We got into it in a very roundabout way several years ago, in fact, in 2012, when we were interested in identifying microRNAs, pathogenic ADPKD microRNAs. We did this screen where we looked at microRNAs that are apparently expressed in murine and human ADPKD samples, overlapped the 3 data sets, and came up with a common list of aberrantly expressed microRNAs. And in it, we found a microRNA called miR-17. We've stuck with this microRNA for all these years, published quite a bit on it as well. And the reason we stuck with this microRNA, many reasons, but one of the top reasons is that just a cursory search of what miR-17 targets leads us to this, that PKD1 is a miR-17 downstream target. I'm showing you a snapshot of a PKD1 3' UTR nucleotide sequence in humans aligned with other species. And the red box indicates the miR-17 binding motif. Now, this was a bioinformatic prediction at the time, but we've proven that this is true biologically as well. Indeed, microRNA 17 physically binds to this region in PKD1, and that this binding represses PKD1 activity using induciferous assays. So the hypothesis then became that in ADPKD, where one PKD1 copy is already mutated, mRNAs made by the remaining copy are inhibited by epifactors, in our case, miR-17, further lowering gene dosage, leading to ADPKD progression. So we've attempted to test this axis in as many ways as we could think of using in vivo models. So I'm going to show you a few examples. First experiment that we did was we transgenically overexpressed miR-17 in normal mice in renal tubules that primarily collecting ducts. And as you can see from H&E and DBA, which labels a collecting duct immunofluorescence here, that miR-17 overexpressing mice, kidneys from these mice have tubule dilations and cysts. To go along with this histological phenotype, at the molecular level, we found that high levels of miR-17 were associated with reduced PKD1 mRNA expression. So in summary, what we found here is that miR-17 upregulation seems to do two things. One, produce a PKD-like phenotype, and two, reduce PKD1 gene dosage in vivo. So it provides some level of sufficiency, if not complete sufficiency, maybe partly sufficiency, part sufficiency in the PKD process. So the next question obviously was a necessity. And so for that, we needed a reduced PKD1 gene dosage model. And for that, we turned to Peter Harris and Kat Hobb from the Mayo Clinic, at the time she was at Mayo Clinic, and used our PKD1 RC mouse model. And we made a minor adjustment to the model, and that's shown here. So one allele of the PKD1 gene in this model has the missense RC mutation in the germline. The other allele is wild type, except that it contains LOXP sites flanking in the introns flanking critical exons of PKD1 gene. Now, in this setting, we introduce a kidney-specific CRE, and that obviously leads to the deletion of the FLOX allele, and in essence, producing a monoallelic PKD1 RC minus mouse model. Indeed, in this setting, PKD1 gene dosage is reduced, and it leads to a reliably aggressive cystic phenotype. So this is a kidney from an 18-day-old mouse, and as you can see, it is virtually destroyed because it's filled with cysts. Obviously, miR-17 is activated in this model, in these kidneys. In this setting, when we delete miR-17, we see a profound suppression of PKD growth, shown here using histology, but also kidney weight body weight ratio and BUN levels go down to near normal levels. What's more, PKD1 dosage goes up when in the double knockouts compared to the single knockouts. So simplistically speaking, there are some caveats to this, but simplistically speaking, the only way PKD1 could go up in the double mutants is if the remaining RC allele made more mRNA, which led to more PKD1, polycystin 1 protein. So this data to us suggests miR-17 necessity, at least in this model, for ADPKD progression. And it brings me to a very simple interim model, straightforward. We all know PKD1 is essential for repressing cyst growth. Based on this data that I just showed you, miR-17 represses PKD1, modifying gene dosage, and maybe playing a role in that way in aggravating ADPKD progression. But biology is not this straightforward or simple. There are issues with this model. First, I cannot draw a straight arrow between miR-17 and PKD1 because all we've shown so far is association. Every time miR-17 goes up, PKD1 goes down, or miR-17 goes down, PKD1 goes up. It's an association, not a causation, at least in vivo. Two, we know that microRNAs are not like transcription factors on and off switches. They function as rheostats, and they subtly repress their gene targets, 20–30% repression. Would that level of repression be sufficient to have any disease relevance in this aggressive model? And more importantly, miR-17 has tens, if not hundreds of other targets, even in the context of PKD. What's to say that these other targets are not contributing to ADPKD pathogenesis? So how do we disentangle and decouple this pleiotropy? In other words, we need to devise an experiment where miR-17 is not deleted. It's actually allowed to continue its repressive activity on all its mRNA targets except PKD1. If this scenario recapitulates the effects of deleting miR-17 entirely, then you can prove PKD1 as the causative link in this equation, and also that the mRNA was indeed repressed at a biologically relevant way. So to do this, we use this approach, CRISPR gene editing. So here I'm showing you a schematic of PKD1 gene. At the tail end, the blue box is the UTR, which contains the red box, which is miR-17 motif. Now, the mRNA that is made by this gene obviously contains the miR-17 motif in this 3' UTR as well. So we designed CRISPR guides to delete this motif. And so the idea is that now we have a new gene that lacks the miR-17 motif in its 3' UTR, and the mRNA that will be made by that gene will lack miR-17 motif, preventing miR-17 from binding to this mRNA, only to this mRNA, and then we want to study the effects of this manipulation in vivo. So here we've modeled this in multiple different systems, in normal cells, normal mice, ADPKD models. Here I'm just showing you an example of one of those modeling where our system or approach seems to work. The green bold nucleotides are the miR-17 motif. The scissors are the guides, and we delete the green and some flanking red regions. The free ends then reconnect, making a new 3' UTR that lacks the miR-17 motif. When you model this deletion, I'm not going to show you the data. When we model this deletion in normal cells or mice, we find that the gene behaves just fine. Mice are fine, no cis, so the gene does not get inactivated. So in other words, you can muck around with the 3' UTR, and it does not seem to affect the translation of the mRNA. What I want to show you, though, is modeling of these mutations, of these deletions in the context of ADPKD. So here is the experimental setup. I've already shown you this model, the missense RC null a mouse, and I've shown you that this produces aggressive polycystic kidney disease. We would propose that the remaining allele has an Achilles heel, which is its miR-17 binding site in its tail end. We designed a parallel experiment where we now removed that binding site, that Achilles heel, and the question is what would happen to polycystic kidney disease in this setting. So here are the data. The nomenclature may get a little confusing, so please feel free to ask me in the chat or afterwards. But here is the PKD1 RC plus kidney, which is normal. One allele has RC, the other allele is wild type. This is the RC minus kidney, which I've already shown you before, and that gets PKD. Now, when we remove the miR-17 binding site from the RC allele, and the other allele is still wild type, the kidneys are fine, just like the RC plus mice. So deleting miR-17 binding motif from the RC by itself does not do much. In contrast, when you remove the the miR-17 binding site from RC allele, and the other allele is now deleted, we see profound benefit in terms of reduction of cis growth. I want to mention upfront that all of these 4 different genotypes were derived from the same founder, so founder lines. So these are genetically as genetically identical as possible. So these differences are not because of some changes in genetic background. What's more, we find that compared to the RC minus, RC delta 17 minus mice have higher PKD1 polycystin-1 expression levels. So we've measured polycystin-1. Everybody here knows polycystin-1 biochemistry is not that straightforward. We're showing you here is 462 kilodalton band using the 7E12 antibody that detects the full length and the N-terminus. We've confirmed these polycystin expression levels using a second independent antibody that's produced by the PKD-RRC, University of Maryland core, and that detects the CTF. So this happens, we've done this experiment a bit more rigorously. This is founder 1. We did this in a second founder, founder number 2. Again, we see the same sort of a phenotypic regression, and founder number 3. There are also differences in each of these founders. So founder 1, for example, has just has a 53 base pair deletion that contains the miR-17 motif. Founder 2 has a 108 base pair deletion in the 3' UTR that also contains the miR-17 motif. And founder 3 is just an 18 base pair net loss from the 3' UTR that also contains the miR-17 motif. This from a 3' UTR target standpoint, it tells you where the money really lies. We've gone from 108 to 53 to 18 base pair, all of which containing the miR-17 motif. Okay, so next, is this a phenomenon of the RC mouse, or is it more generalizable? And so for that, we've turned to primary ADPKD, human ADPKD cell lines. And so these are cells that we obtained, again, from the PKD-RRC, this time the University of Kansas, graciously provided by Dr. Darren Wallace. And he sent us cell lines from 4 donors. We did a couple of things with it. First, we genotyped the cis cells. And we find that 3 of the 4 donors have pretty bad PKD1 mutation, and the other one has a PKD2 mutation. But please note that these cells came from Cis, and we did not see a second hit. The other allele of PKD1 or PKD2 was still present. So next, what we did was we divided each of these cell lines into 2 groups. One in which miR-17 motif was deleted using guides that are specific to the human 3' UTR miR-17 binding site, or isogenic controls where miR-17 binding site was left intact. And in each case, we find that deleting the miR-17 binding site leads to upregulation of polycystin-1 protein. Again, this is 462 kilodalton protein that we're looking at here. When we grow these cells in 3D cultures, matrigel 3D cultures, we find that the primary ADPKD cells that lack the miR-17 binding site and have higher PC1 expression levels form smaller cysts in this assay.
3 minutes.
I'm almost done. So this, I hope, tells you or convinces you that it provides a basis for targeting the miR-17 PKD1 axis as a drug target axis for ADPKD. And one of the ways of thinking about it is perhaps developing a drug that turns off miR-17, an anti-miR-17. And make a long story short, that is indeed what we have done. And here I'm showing you some data about that. We went back to the model that I just showed you, the RC minus mouse, and treated it with our anti-miR-17 drug here called RGLS4326. And compared to mice that got PBS or control oligonucleotide, the RGLS4326 treated mice had profound benefit again in PKD, pathogenesis, kidney weight body weight ratios, went almost back down to baseline, and creatinine was normalized at this time point. So what I've shown you so far is that 3 ways that this system seems to be that we have manipulated. 1, by deleting miR-17. 2, by deleting the miR-17 motif only from the PKD1 3' UTR, and 3, by pharmaceutically inhibiting miR-17. And in each case, we find that PKD1 levels go up, and there is benefit from a ADPKD standpoint. This drug has been tested in a phase 1, a small phase 1 trial, and I'm showing you a top-line data from that here. So these were 9 subjects with ADPKD, class C, D, or E, which is aggressive, relatively aggressive ADPKD, and what the investigators did was they measured urinary polycystin-1 levels at baseline, which is before the drug was given, and at various stages after the drug was given. There were 4 doses spread out every other week. And what you can see here is that compared to baseline, urinary PC1 levels, urinary exosomal PC1 levels go up in all of these 9 subjects after drug treatment. This too suggests that one, from a drug development standpoint, that the drug actually delivered to the kidney, but 2, from a biology standpoint, that perhaps the miR-17 mediated repression of PKD1 may be irrelevant even in ADPKD patients. So this brings me to my conclusion. I conclude with this beautiful paintings of Achilles, and we propose that PKD1 remaining allele in the context of ADPKD has an Achilles heel. It's 3' UTR miR-17 motif, which leads to its cis-suppression, lowering gene dosage and aggravating ADPKD. The cool new tool would be drugs that harness this mechanism and activate or de-repress the remaining PKD1 allele. So with that, I'd like to acknowledge the people that did the work. in particular, people shown here in bold, Dr. Ronak Lakia and Harini Ramalingam, led the recent 3' UTR studies and are co-first authors on that paper. And key collaborators, Edmund Lee from Regulus Therapeutics and Dr. Darren Wallace from the University of Kansas, and my funding. And with that, I'll stop and take questions if there are any.
Okay, yeah, thank you very much, Dr. Patel. I think it was really exciting new approach. So let me see if we have questions in the chat. So if anybody has questions, please write in the chat so that I can read. So let me start with my quick question. So I think the microRNA has multiple targets. I think in this case, microRNA 17 should also have some other target genes. When you use this anti-microRNA 17, do you expect any off-target gene regulation that could cause some kind of side effects in the patient?
Yeah, so I mean that's an obvious question. The anti-microRNA 17 does the good thing, which is it raises PKD1, but you're right, it may raise other MIR17 targets as well. I think the way that the drug did, there are ways of hedging against off-target effects when it comes to drug development. One is to make it as targeted as possible to deliver this drug to the targeted cells, not to that's extra-renal tissues. Indeed, that's what the chemical design of this drug is. Indeed, what makes this drug do. It makes it preferentially go to the kidney over other tissues. Now we have some really nice data showing that. So that's maybe one way of seeing that you don't run into off-target effects. The other thing is that hundreds of predicted targets, but they may not be co-expressed in the same cells to which the drug is going. So it's not going to de-repress all of these 1,000 targets. And then finally, when you delete MIR17 in adult mice using inducible germline whole body deletions, the mice are fine, meaning they're fertile, nothing happens to them. The only effect they have is the hematopoietic stem cell population, the turnover becomes lower. And so as long as the drug does not go to that population based on the mouse genetic data, you would predict that there will be off-target effects. But that I can't. But obviously you don't know until you do the experiments.
Right.
So
yeah. Thank you very much. So we have many questions now. So let me start from the first one. So can the RGLS drug decrease the other microRNA 17 family members expression?
Yes, so the drug is designed, it's an I-nucleotide drug, which is designed to base pair against the seed of the entire MIR17 microRNA family. And so, yeah, it simultaneously inhibits all MIR17 family members, not all MIR17 cluster members, because the cluster contains microRNAs that belong to other microRNA families, but the MIR17 family members, yes, all of them get inhibited.
Okay, so the next question is from Dr. Paul. So is there a biology around microRNA 17? Is it, for example, regulated to regulate PKD1 physiologically or in development? So,
yeah, good question, Paul. So what we know of what regulates MIR17 is there's actually pretty convincing data that c-Myc is an upstream activator of MIR17 transcription. There is a syndrome called Feingold syndrome, which is caused due to mutations of, a developmental syndrome. It's caused due to mutations of MYC. Some of these Feingold-like patients do not have mutations in MYC, and there was a Nature Genetics paper several years ago that found that those were due to homozygous miR-17 deletions. So even in humans, this MYC miR-17 axis remains intact. In PKD, we've proven that miR-17, when you transgenically express MYC, 17 goes up. When you delete 17, MYC does not go down. So, it really is downstream of MYC. We've done CHIP assays and things like that to prove in vivo that MYC binds there. So MYC, I think, is the upstream target, upstream activator, and that puts miR-17 into the developmental realm of its physiological function. So if you delete miR-17 cluster during embryogenesis, it actually leads to perinatal lethality because of developmental defects in the hematopoietic system, lung, and heart. So that is perhaps its physiological function. Later in life, just like MYC, it may serve as oncogene or developmental repair, activating developmental repair processes and things like that. In fact, there is a study showing that miR-17 appears to go up after AKI. So yeah, that may be its developmental function.
Okay, let me go to the next question. So the next question is, did you test if expressing some unrelated mutant protein, something misfolded that might trigger ER stress pathways, would have some effect on PKD?
Yeah, well, we haven't tested any misfolded proteins. I mean, the RC mutant was thought to be was thought to muck up the ER, and obviously here we didn't see that. So, but yeah, we are, and all the other PKD mutations that we've tested this approach in appear to be inactivating monolithic inactivating mutations. So, but yeah, we are in search for other proteins. Yeah.
Okay, so the next question is, have you tried the drug microRNA 17 in other models different from the RC?
Yes, we've done it in PKD1 nulls, PKD2 nulls, in the PCYs, the KIFs. Yeah, we've done it in a bunch of other places. And the most profound benefit is obviously in this model because that's where PKD1 is still present in the PKD1 nulls. It seems to work, but not to this degree, suggesting, and there it may be working through downstream pathways. One of the pathways that we've implicated is PPAR alpha mitochondrial metabolism pathway. Yeah.
Okay, so the next question is, your plots look like PKD1 is also upregulated in wild type mice. So is this just a background tonic repression by microRNA 17, or is there truly regulation in the sense that there are physiological, pathological drivers of microRNA 17?
Yeah, this is it's from Alan, right? I can talk to him off some things, but we did see that, but it was not a consistent feature, and all the founders or all the time every time we tested that, and then especially when we aged these mice, let's say P28 or older ages, we didn't see the de-repression of PC1 that you're mentioning. So yeah, whether so in my mind, the miR-17 PKD1 regulation in the physiological context is still unclear to me. We haven't figured that out. In the PKD context, it's pretty clear, but in the normal context, we haven't seen a consistent signal.
Okay, yeah, so thank you so much. So we have to be on time. So there are several questions. If you can answer in the chat, that would be great. So let me introduce.
Thank you so much.
Thank you so much. So let me introduce next speaker. Is Dr. Christine Knowles. She is the Whitehead Career Development Professor in the MIT, and she graduated Harvard MIT MD PhD program, and she started her lab in 2018 at the Whitehead Institute. And her lab develops high-throughput functional genomic approaches to understand and modulate organ injury and regeneration. Dr. Knowles, can you share your screen?
Thank you so much. Good morning, everyone, and thank you again to the organizers for including me in this meeting. Over the next several minutes, I'm going to take us out of the kidney and into the liver to discuss our lab's work to bring high throughput functional genomics into mammalian tissues. And I'll end by discussing how we might be able to bring this into other tissues like the kidney. And so our interest in developing this technology really emerged from our desire to answer a fundamental biological question, which is what underlies the unique regenerative ability of select organs, and can we somehow confer this to other tissues in the setting of disease? And so if we think broadly about the regenerative ability of different organs in our body, we largely classify them into two distinct categories. There are those like the intestine and the hematopoietic system that can continuously renew, owing to the presence of stem cells that can constantly replenish those terminally differentiated cells responsible for organ function. And then there are those like the heart and brain that cannot renew, as they're comprised entirely of terminally differentiated cells. And so a longstanding and still elusive goal in regenerative medicine has been to somehow stimulate the endogenous renewal and repair of these organs that lack stem cell populations. And that's where the mammalian liver really emerges as a remarkable and I think highly informative exception. Because while the liver has very low turnover at baseline, it has the ability to completely regenerate itself after injury. And importantly, this regeneration is driven not by a stem cell population, but rather by the proliferation of these differentiated hepatocytes. And so these differentiated hepatocytes are considered to be quiescent rather than terminally differentiated. And so this exceptional regenerative ability of the liver tells us that in principle, organ regeneration is possible without a stem cell population. But we still have no idea why it is that hepatocytes can proliferate and therefore the liver can regenerate, while other differentiated cell types and the organs in which they reside cannot. And so if you think about how we might answer this question and really any question pertaining to tissue biology, we can certainly take these cells out of the organism and study them in culture, where we'd benefit from tractable experimentation and the ability to make both observations and perturbations in a high throughput manner. But in bringing these cells out of the organism, we necessarily strip them of the many things that naturally regulate their behaviors. This includes interactions with the extracellular matrix and other cell types, the influence of circulating factors, as well as the role of biological sex. And so for processes occurring at the tissue scale, such as regeneration, which we already know to be subject to regulation by such factors, and as this group can very much appreciate, it's really important that we study these processes in their native context in order to capture the complexity of this regulation and achieve a very comprehensive and ultimately therapeutically relevant understanding of this process. But studying processes directly in the organism has long required that we sacrifice some experimental tractability, because while we can certainly still perform high throughput observational experiments like transcriptomics and proteomics, when it comes to inferring causal relationships, we're largely restricted to analyzing the function of single genes at a time through knockout mice. And this has really limited our capacity to understand mammalian physiology and disease in a comprehensive and unbiased manner. And so it's for this reason that I was really keen to try to bring accessible high throughput functional genomics into the liver, and thereby bring that experimental tractability into a physiologically relevant system. And so within the functional genomics space, genome-wide screening using CRISPR-Cas9 has really emerged as a powerful tool for uncovering the complete regulation of cellular phenotypes. The original approach involves lentiviral delivery of a genome-scale guide RNA library alongside Cas9 to cells in culture. And typically this means delivering tens of thousands of guide RNAs to tens of millions of cells to achieve sufficiently high coverage such that you can ultimately detect depletion and enrichment of guides and thus hits in the screen by deep sequencing at the end of selection. And so this overall approach has since been applied to hundreds of different cell lines to uncover the regulation of diverse phenotypes in cell culture. Beyond cell culture systems, people have also been able to introduce guide RNAs to cancer cells or immune cells ex vivo, and then inject these cells into animals to allow for phenotypic selection directly within the organism. And this has allowed for high throughput dissection of phenotypes in the setting of cancer xenografts and the immune system. But when it comes to dissecting phenotypes within tissues, we just have not been able to deliver lentivirus into these tissues at the efficiency required for genome scale screening. And this has required the use of other delivery tools that have significant limitations. And what this has meant practically is that thus far, genetic screening in tissues has largely been limited to small scale positive selection screens on the order of tens to hundreds of genes. And so there thus remains an unmet need to be able to perform high throughput positive and negative selection screens in organismal tissues. And this begins with us finding a way to get lentivirus or other stable delivery methods into these tissues. And so after several months of trial and error in the mouse room, I found that if I injected lentivirus intravenously into a neonatal mouse, I could actually achieve efficient and rather stable transduction of hepatocytes. And so we designed a very simple lentiviral vector in which we can express a guide RNA as well as a fluorescent reporter. And to get a sense of transduction efficiency and the multiplicity of infection, I mixed equal amounts of an mCherry and mTurk2 lentivirus, then concentrated this lentivirus about 1,000-fold and injected between 2 and 50 million TUs intravenously into postnatal day 1 mice. And what you can see here is that we can actually transduce a wide range of hepatocytes in a dose-dependent manner, shown here in these low magnification images of livers harvested 4 days after injection. And the data are quantified here, where you can see that at a dose of 50 million TU, we're able to transduce over 75% of hepatocytes. And at this dose, based on the frequency of single and dual color cells, it appears that we have an MOI of approximately 2. And importantly, if I injected a set dose into mice and then looked at these livers either 4 days or 14 weeks after injection, I found that these transduced hepatocytes were indeed persisting throughout the lifetime of the animal. Now, more broadly, given that there are approximately 13 million hepatocytes in the postnatal day 1 liver, it means that at this highest dose of 50 million TU, we're transducing approximately 10 million hepatocytes at an MOI of 2, which means that we could actually achieve about 200-fold coverage of your typical genome scale guide RNA library in just a single mouse liver, which actually puts us within the coverage required for genome scale screening. So the next question then is, can we use this lentivirus-based approach to deplete proteins of interest in these hepatocytes, even when they may not be undergoing the rapid population doublings associated with cells in culture? And so to test this, I settled on targeting the mitochondrial enzyme MAO-B, as it's a non-essential protein with a long half-life, and therefore can serve as an upper bound for the kinetics of protein depletion. And to achieve temporally controlled induction of Cas9, I took advantage of commercially available lock-stop-box Cas9 mice, in which we could induce Cas9 in all hepatocytes via intraperitoneal injection of an adeno-associated virus expressing CRE, henceforth referred to as AAV-CRE. And so to test Cas9 induction and protein depletion in neonates, where hepatocytes are still proliferative, I injected lentivirus encoding a guide RNA targeting MAO-B alongside mCherry into postnatal day 1 mice. And then 4 days later, postnatal day 5, I injected either PBS or AAV-CRE, and then harvested 1 and 2 weeks thereafter to look at MAO-B levels in single cells. And as you can see, in the absence of AAV-CRE and Cas9 induction, we don't see any depletion of MAO-B, even in cells expressing the guide RNA. However, in livers in which we've induced Cas9 with AAV-CRE, we see the complete and specific depletion of MAO-B exclusively in cells expressing the guide RNA. And this is complete after about 2 weeks. And so this is a nice confirmation that our system works as expected. And it tells us that in the neonatal liver, we need to wait at least 2 weeks after Cas9 induction for any sort of phenotypic selection to take hold. Now, importantly, in neonatal mice, hepatocytes are still highly proliferative as part of liver growth. And so a subsequent question and an important question is, can we still achieve protein depletion in an adult liver where the hepatocytes are quiescent and not proliferative? And so to test this, I repeated the experiment I just described to you, except that I waited until the mice were 8 weeks of age to induce Cas9 and then harvested the livers 2 and 4 weeks thereafter. And so indeed, as in the neonate, you can see that we do not get any ectopic MAO-B depletion, even when these cells have been expressing the guide RNA now for several weeks. However, again, in the setting of Cas9 induction, we get the specific depletion of MAO-B in the mCherry positive cells. And this is, again, complete between 2 and 4 weeks. And so this combination of lentiviral-mediated guide RNA delivery and AAV-CRE-mediated Cas9 induction really offers an effective platform for hepatocyte-specific protein depletion and phenotypic selection at any point in the animal's lifetime. And so with this efficient means of delivering guide RNAs to the liver and inducing Cas9, we felt comfortable moving forward with a genome-wide screen in the liver. And so to do this, we elected to do a screen for hepatocyte fitness in the neonatal mouse liver, because there we have a wealth of disease data in humans to validate our findings, as well as hundreds of fitness screens and cell culture in which to compare our results. And so to do this, we injected a genome-wide guide RNA library, in which we have guide RNAs targeting genes across the entire genome, into 4 male and 4 female inducible Cas9 mice. At postnatal day 5, we harvested livers from 4 of these mice to serve as the induction time point for the screen. And then we injected AAV-Cre into the remaining 4 mice to induce Cas9 and allow for subsequent selection. Now, we then harvested these livers 3 weeks later at postnatal day 26 to serve as the endpoint of the screen. And we know that over this 3-week period, hepatocytes are undergoing approximately 3 population doublings. And so the screen is testing for the ability of hepatocytes to both persist and proliferate in the growing liver. And so after this 3-week period, we sequenced all of the livers in this screen. And then we calculated the log 2-fold change in guide RNA abundance at endpoint relative to induction. And we call this calculation a gene score. We calculate this for every gene in the genome. And we did this for each individual mouse. And so here you see the results for one of the male mice at endpoint. And when we applied statistical cutoffs to these gene scores for just this one mouse and for all the other individual mice, we found that we could actually identify both significantly enriched and depleted genes in just one mouse. And so the next question was, well, are these results at all reproducible across mice? And as you can see in these pairwise comparisons of gene scores across mice, we indeed observed a very strong correlation in the gene scores between any given pair of mice at endpoint. And together, these results tell us that we can actually perform a genome-wide screen in a single mouse liver, and then easily scale that screen up across additional mice to provide the power of what is truly a biological replicate. And so for subsequent analyses, we combined the gene scores across all 4 mice, and this gave us the statistical power to identify an even greater number of hits. And so we next asked, well, is this screen at all reliably reporting on regulation of the phenotype in question? And in this case, the ability to have hepatocytes to both persist and proliferate in the growing liver. And so to test this, we didn't want to make any assumptions about what we know to be required for cell fitness in culture. And so we instead turned to human disease data, where we know that disruptions of specific genes is known to affect hepatocyte fitness. And from this, we compiled 2 lists of genes. The first is a set of genes known to be mutated in neonatal liver failure syndromes that are required for hepatocyte viability. And so we would expect the guide RNAs targeting those genes, and thus the genes themselves, to deplete in our screen. The second is a list of genes documented to act as tumor suppressor genes in hepatocellular carcinoma. And so you would expect that guide RNA-mediated knockout of these genes would cause those cells, and thus the guide RNAs, to enrich in our screen. And so we would call those genes as enriched. And so we then look to see how these genes actually performed in our screen. And so here you see a volcano plot of the compiled gene scores across the 4 mice. And when we looked at the performance of these the genes in these 2 sets, you can see that indeed the genes we expected to deplete in our screen were significantly depleted, and the genes we expected to enrich were significantly enriched. And so this told us that our screen actually had the ability to reliably uncover regulation of hepatocyte fitness, and importantly, that we could observe both depletion and enrichment of genes in a matter of just a few population doublings. And so we next did a gene set enrichment analysis of all of the genes depleted in our screen. And not surprisingly, we found that many of the depleted genes were belonging to pathways classically associated to be required for cells in culture. And indeed, a list of core essential genes that has been annotated based on fitness screens of cells in culture was significantly depleted in our screen. And so we find that, not surprisingly, cells in the organism share many of the requirements to cells in culture. But as I mentioned at the beginning, the whole motivation for developing this method was to identify the broader regulation of phenotypes that we can't reproduce in cell culture. And so to see whether we could uncover any such regulation in this pilot screen, we looked to see if there were pathways that were uniquely depleted in our screen relative to cell culture screens. And so to do this, we compared our screen to 2 mouse embryonic stem cell screens to thereby control for any species-specific requirements for cell fitness, and also to 24 human hepatocellular carcinoma cell line screens to thereby control for any lineage-specific requirements. And when we performed pathway enrichment analysis, we found that there were 4 gene sets that were uniquely depleted in our screen relative to all of these cell culture screens. These pathways were protein export, vesicular transport, antigen presentation, and glycosaminoglycan biosynthesis. And when we looked at the genes in each of these pathways, we found 2 trends. For protein export and vesicular transport, we found that these genes were actually depleted in all screens, but to a much greater extent in our screen. However, for antigen presentation and glycosaminoglycan biosynthesis, we actually observed something quite different. For both of these pathways, we observed that there were genes that were depleted in our screen that never exhibited depletion in the cell culture screens, indicating a unique requirement for these genes and pathways in the living organism. For antigen presentation, we found that genes involved in the class 1 MHC pathway were absolutely essential for hepatocyte fitness in the organism, possibly to protect against immune-mediated clearance. And for glycosaminoglycan biosynthesis, we found that genes specifically involved in the synthesis of heparan sulfate were absolutely essential in the living organism. And we hypothesized that this might be due to a requirement for heparan sulfate in the extracellular matrix and on membrane proteins to actually help recruit and potentiate growth factor signaling for individual cells. And so to summarize what we've established and what I've just shown you, within the liver, we have now created an approach that is comprehensive. You can query the entire genome through both positive and negative selection, and it's highly adaptable. We can induce Cas9 at any point in the animal's lifetime, and provided you have a means of enriching or staining for your favorite phenotype, you can now select for it. And you can imagine adapting this overall approach I just presented to the various other CRISPR-based applications available. And finally, it's an accessible method. It requires similar reagents to cell culture screens, minimally requires just 1 mouse, and can be easily scaled across mice to provide the power of true biological replicates. And so I'm really excited for this approach to open up an area of organismal functional genomics and bring that experimental tractability that was once restricted to cell culture systems into animal models, so that we can now achieve deep molecular insights into these diverse aspects of mammalian physiology and disease.
3 minutes.
And so for us, we're now leveraging this tool to dive back into those fundamental questions of cellular quiescence and organ regeneration that I posed at the beginning. We're now performing screens in the adult liver to identify the genes required for dormant cells as opposed to proliferating cells. We've since adapted the screening approach to screen for single cell phenotypes, and we're now performing screens to identify those genes that endow the liver with its remarkable regenerative ability. And lastly, we're working to bring this technology into other organs for us to identify barriers to regeneration in those tissues, but also to just broaden the utility of this technology to anyone interested in understanding physiology and disease. And so given this audience, I'd just like to end briefly by expanding on what it would take to bring this into other tissues. And so the technical challenge really can be distilled down into 2 key components: guide RNA delivery and guide RNA coverage. On the delivery side, in order for guide RNA representation to be evaluated at the end of screening, these guides have to stably integrate into the genome of the target cells. And so it's for this reason that lentivirus is a natural choice. But in our hands and in others, it seems that standard VSVG pseudotype lentivirus, while it can be targeted to the liver if injected in specific ways, we find that we cannot get it to target efficiently to other tissues. And so we're now pursuing lentiviral envelope engineering approaches to try to retarget lentivirus to other cell types in the organism, and others are pursuing similar envelope engineering approaches as well. Now, alternatively, we could turn to other targeted delivery vectors, but importantly, a lot of the delivery tools out there that do go to other tissues only result in transient expression. It would not allow those guide RNAs to integrate into the genome. And so we would need to couple these delivery vectors with integration-based machinery. And then on the coverage side, in order to reliably detect changes in guide RNA abundance over the course of screening, you really need to have each guide represented by between 250 and 1,000 cells. And so for a genome scale library, this means then having those guide RNAs in tens to millions of cells. And so for some tissues, efficient delivery of guide RNAs will be sufficient. But for other tissues where we simply don't have that number of cells, we'll have to think about creative ways to reduce the number of guide RNAs needed per gene, and also, or alternatively, to reduce the number of cells we actually need per guide RNA. And so to end, I'd just like to acknowledge that this entire approach was developed while I was a fellow at the Whitehead Institute, and it was a wonderful collaboration with Heather Keyes, who runs the Institute's Functional Genomics platform. And I'd also like to thank the various core facilities that helped at various points in that endeavor. And finally, I'd like to say that I recently transitioned to a faculty position at MIT with our lab based at the Koch Institute. And so I want to thank my lab members for being incredibly brave and enthusiastic in taking on the new directions in our lab. And we are also very keen to have people join us in these endeavors. So if any of this piqued your interest or you know of anyone who might be interested, please do spread the word and connect them to us. So thank you so much for your attention, and I would be happy to take any questions.
Really cool tools, Kristin, and excellent talk. Thanks a lot. So the first questions are coming in already, and I just read them. So the first one is, I may have missed that, but how did you target liver in mice?
So we found if we inject highly concentrated lentivirus at the dose we're injecting is about 50 million TUs. So it's about 1,000-fold concentration from what we're typically harvesting in supernatant. If we deliver that intravenously into the neonate, there's a facial vein that is visible and accessible in a postnatal day 1 mouse. If we inject it intravenously, we do get that efficient delivery to the liver. Unfortunately, if we look at other tissues, we do not see as efficient delivery. So it seems that this approach works very well for liver, but not so much for other tissues. And so that's why we're starting to now look into other approaches and actually modifying the lentivirus itself.
Question from Michael Kaplan. So beautiful talk. I'm wondering how the heterogeneity of cell types represented in the kidney might complicate the application of your elegant approach.
Yeah, it's a great question. So we were a little bit spoiled and had quite a bit of a low-hanging fruit in the liver, in that you could, if you want to be incredibly simplistic about it, you could argue the liver is just a bag of hepatocytes, and in fact, tens of millions of hepatocytes. And so when we think about moving this approach into other tissues, we confront the fact that there are often way fewer cells, and then if you distill that into specific cell types, even fewer cells of a particular type. And so I think there, as I sort of alluded to at the end, it's a question of you have to somehow still get that necessary coverage to make inferences. And so then it's a question of, do you want to scale down the library itself and maybe go subgenome, or do you want to try to, I think there are efforts emerging of trying to reduce the number of guide RNAs needed per gene, and also perhaps the number of cells needed per guide. And you can also think about doing a few more guides per cell. So there are ways to reduce it, but if you're going to go down orders of magnitude from tens of millions of cells, I think it would require some compromises on library. Or you can certainly scale up mice. So I don't have a single answer to that, but I think there's different approaches one can take to try to bring the numbers into the realm of possibility.
Next question is, do you know how efficiently you could transduce cholangiocytes? That's the right word. Sorry for that.
Yeah, so right now we're actually, we believe, as I mentioned, our lentivirus, our fluorescent reporter we were using is a hepatocyte specific promoter. And then the screen itself is hepatocyte specific by virtue of us inducing Cas9 only in hepatocytes. We are now looking to see whether we can get lentivirus efficiently to cholangiocytes in the liver. And if not, we're going to, as we now move into this envelope engineering approach, we're going to start looking into targeting the other cell types in the liver, because there's a lot we would like to start studying about those cell types. But while it's while the liver's famous or infamous for being easily targeted, is really the hepatocytes. And so getting to those other cell types would certainly be important.
And that's a similar question just for the kidney. Would it be possible to design a panel that targets a certain set of genes and deliver to a certain cell type in the kidney? So it's
to design a library.
So basically a set that only targets a certain set of genes and deliver to a certain cell type. So gain more specificity and not do the genome-wide approach, basically.
Yeah, that's absolutely possible. And I think, you know, in the liver, we could do genome-wide, so we did. But of course, the vast majority of genes are not essential or not relevant. The question is, how do you know that up front to preserve the unbiased nature? I think things that can be done is you could consider looking at in vitro data for, you know, kidney derived lines and see what genes are essential or not essential and either eliminate or include them accordingly in the screen. That can actually reduce libraries by significant fractions. It's also possible to just do subscale libraries. You can, in where the technology is at, you can simply put a list of genes into the Broad Institute's Guide RNA website and design as many guides as you want for any number of genes, anywhere from 0 to 20,000. And so if you have some sense of what may or may not be important, you could absolutely do that. In another thing you can also do is pour a lot of effort into a genome scale or high throughput screen, and then that gives you a really nice foundation of what is essential and what is completely neutral. And then for essential genes that you know are always going to be important, you can almost drop them out to then do any other specific phenotypic screening. And so that's one thing we've also done moving forward, is we just get rid of all of those essentials because we know that the cell is going to die. And so if we're interested in anything else the cell does, it's less useful.
I see. Just a very short last question. So a permutation of Michael's question: could your approach allow delivery of mTOR to kidney liver epithelia and help us with the challenge of generating immortalized cell lines?
In vivo or?
Yeah, so basically doing that in vivo and then getting the cells out in the end.
Yeah, so we have not specifically looked at lentiviral targeting to kidney yet. We can certainly look and see whether our IV approach goes there. I do not expect it to, because in the other organs we've looked at, we do not see efficient delivery. But one thing we are working on right now is an unbiased screening platform to look at a broad range of lentiviruses displaying various targeting peptides and see where they go. And so that will hopefully allow us to, the goal would be to find a specific targeting peptide for any cell type in the body. And the hope would be that we could get that there. But right now we have not seen any efficient delivery to kidney, but we have not looked closely.
All right. Thanks a lot, Christine, again. And we move on to the next talk. So the next talk is by Chung, who is an associate professor in the Department of Biomedical Engineering at the University of Southern California. And her research focuses on design and application of self-assembling multimodal micelle nanoparticles for theranostic engineering. And today she will actually present us the peptide-based micelles for PKD. So
all right, thank you so much for that introduction. Are you seeing my slides okay? Look good?
Yes,
on
all
your
good,
end?
all good,
Okay,
yes.
Perfect. All right. So yeah, thank you to the organizers and the moderators for putting on a great symposium. And I'm happy to take part of it and share with you, I guess, a lot of the tools that we're making as part of our lab's research. So I'm going to be talking about these peptide-based nanoparticles that we call micelles for polycystic kidney disease. And I thought I'd orient everybody a little bit about who our lab is. You know, we are a biomaterials and nanomedicine research group. And so that just means that we can synthesize various polymers or molecules and drive them to become an architecture or some sort of molecular assembly to then think about an application. And of course, this is a reiterative process. We're designing and motivated by the ultimate application, always having that in sight. And I'll kind of walk you through a story, right, for PKD and how we're thinking about it in this particular realm. But since this is a very broad audience, I wanted to kind of get everybody thinking about sort of what exists on the nanometer scale and what that actually means. If you think about a human hair, it's about 100 micrometers or 100,000 nanometers. We're going down a fold to red blood cells at about 10,000 nanometers or 10 microns, and nanoparticles exist between 10 and 100 nanometers. Obviously, we can get a little bit bigger than that. Liposomes can be typically around 250 nanometers, but as defined by NIH and various nano-related institutes, we think about nanoparticles about 10 to 100 nanometers or so. So nanoparticles are small, as I was just kind of introducing, but they're actually pretty big compared to small molecule drugs such as chemotherapeutic drugs, which are about 1 nanometer or so. And so that difference between 1 nanometer small molecule drugs and nanoparticles is really kind of that difference between a soccer ball and a Goodyear blimp. And so you can think about how many soccer balls can be stuffed into a Goodyear blimp. And so really the particle is acting like a carrier system and holding a bunch of particles or drugs and can be utilized to carry it to a specific location, which is what we call active targeting. So just to kind of reiterate, nanoparticles are large compared to drugs. And because of that, you can fit multiple drugs and can act as a carrier system, literally carrying these drugs to a particular place. And because of that sheer physical chemical difference, their size or some of their surface properties, they can or cannot access certain parts of the body with more or less efficiency. And so I'll talk about how we're utilizing that kind of approach to drive it to go into the kidney. Of course, though, nanoparticles in general have been around for decades. This is kind of the more common, or let's say the most widely used nanoparticle, or nanomedicine, which is doxorubicin within a liposomal formulation. And it's already been FDA approved since the 90s for various types of cancer. And this particular particle has doxorubicin in the core. It has an ammonium sulfide gradient so that drugs don't get freely out of the particle until it gets to the target site. And this is what it looks like under TEM. And the way that these nanoparticles and this nanoparticle formulation, drug formulation targets the tumor is because within the vasculature, this is, let's say, the tumor vasculature, on the left is healthy vasculature. The endothelial cells have these spaces that is characteristic to tumors. And this is called, and this allows for the particles to actually slip through these physical spaces and enter and get to the tumor site. And this is what we call the enhanced permeability and retention effect, or a way of passive targeting. So the sheer physical and chemical properties of the particle is enabling this targeting ability to get into the tumor site and not into the healthy areas. The nanoparticles for the cancer applications have been around, as I mentioned. It was the liposome in general was sort of formulated in the 60s. And then doxil within liposomes were approved in the 90s. And since then, there were, and before that actually it was decorated with polyethylene glycol to allow for enhanced in vivo circulation. And now, right, we have antibodies on them and different targeting agents for a more, not only a passive targeting ability, but this passive plus active targeting. So it's binding to a very specific receptor and site. And now, since about 3 years ago, nanomedicine has really kind of exploded with COVID vaccines. And so it seems like, lipid nanoparticles and mRNA delivery is something that many of us are now more familiar with than 3 years ago, where there is an mRNA that is a piece that's going to be allowed for translation of a part of the spike protein is within a lipid nanoparticle formulation, and it is ultimately injected in the muscle, allows for cell uptake and processing such that antibodies can be produced by our immune system, and that allows for sort of this protective effect, right? For potentially, if the real thing, if we were exposed to the real thing, we have this type of memory. And the reason why this nanoparticle, though, is important here, and you might ask, why can't we just inject the mRNA just directly into our muscles? Well, it's because the mRNA, we have nucleases, right, that would naturally degrade foreign nucleic acids. And so the lipid nanoparticle enables protection of the cargo. And the second thing is that the nucleic acids won't necessarily get internalized into the cell very well. Whereas when you package it into a nanoparticle, it can not only enable protection, but also then efficient delivery into the cell. But there are these barriers, just like those nucleic acids, to nanomedicine and drug delivery in general. So you might have heard of the blood-brain barrier, let's say, where the brain has this type of privilege, immune privilege. And it also has within the BBB an endothelium that is much tighter in terms of tight junctions than other endothelia that's present in our body. So that doesn't allow for small molecules to slip through and get into tissue, target tissue. Another aspect is that it has a lot of metabolism and metabolic enzymes. And another aspect is related to just these efflux proteins that kind of pump foreign and drugs out. And so that's one potential barrier to, let's say, drug delivery or nanomedicine delivery into the brain. Another barrier to nanomedicine is when you're doing oral delivery, you have this first pass effect where you might take something, a drug orally, it'll have to get through your stomach. So it has to be not degraded in that low pH environment, kind of go into your intestines and then pass the mucosal layer. And then whatever is left behind will then have to pass through the liver, where there is up to 90% of the drugs that get metabolized. And what's left over, 10% or less of that initial drug dose is now available to circulate in the body. And of course, then it has to get to the right place. So a very small fraction of what was original is now reaching the target. Another aspect related to barriers is the reticular endothelial system. So this is just part of our immune system where they see foreign things and things like nanoparticles, and they might put opsonins on them and ultimately sequester them in the liver for degradation. And now nanoparticles will put polyethylene glycol or pseudoRNA materials so that it can hinder protein absorption and antibody tacking on so that it can kind of hinder some of this process. Another barrier is even if it got to the right cell, if it goes down the endosomal lysosomal pathway, it can get degraded. And so you have to have this ability to then escape this entrapment within the lysosome. But now there are sort of these types of trends where we're able to say, hey, you know, depending on the size, this is 1 nanometer to 220 nanometers, there's an optimal size for, let's say, EPR effect for tumor targeting. In terms of solubility, there's some optimal solubility that can enable that, and that can go for charge as well. And I will talk about how we're utilizing charge and as well as size to target the kidney. And so that brings us to our nanoparticle platform, these peptide-based micelles, where it's a very simple platform where you have these peptide targeting agents or therapeutic agents, and you can link it to a phospholipid tail, and you can assemble them to become spherical nanoparticles. You can also make them cylindrical or worm-like or tape-like, depending on how we design these types of individual amplifiers. And you know, we like to think of them as particularly beneficial because they have these degradable and compatible components. You can definitely alter them, add things like polyethylene glycol or other types of polymers to kind of have some tunability in terms of half-life, and therefore, and also add targeting agents so that it can have specificity to the disease site and decrease the off-target systemic side effects. You can add, you know, small molecule drugs that are hydrophobic in the core. You can add multiple peptides, therapeutic. You can add RNA or DNA-based therapies on the outside to make it a combination therapeutic system. And lastly, one of the things that's different about these nanoparticles versus, let's say, liposome is they're incredibly small size. They're on the order of about 10 to 20 nanometers, and that's different from, you know, liposomes or other particles that are more on the 100, 200 nanometers. And because of that, we can enable them to go into the kidneys, and I'll talk about that in the future slides. We have a variety of applications in our group, but of course, I'll be talking about PKD today. And I will be talking mostly about our most mature micelle platform, but we have other types of nanoparticles and other types of targeting agents that hopefully I can share with you in the future. And so this story began when I was actually developing nanoparticles for atherosclerosis, actually targeting MCP1, and that actually might be an interest here as well. And those particles went to the liver, and they're about 15 nanometers in diameter. But I had these other types of particles that were much smaller, and I would, at different concentrations, I was doing a pharmacokinetic studies at different concentrations, at different time points. I saw that they would go to the kidneys very well. And in nanomedicine, oftentimes we think about kidneys as not necessarily a great thing. It's a barrier to nanomedicine because that means it's potentially getting cleared out of the body. But I started thinking about this in a kind of an opposite way. You know, if they're getting to the kidneys anyways, right? And most of our drugs are getting not to the kidneys or a particular place. What if we can enable them to get there, tack on things that will allow them to stay there, and then allow for drug delivery to happen? So, you know, renal clearance, is it a barrier or an opportunity? And if you look at the glomerular filtration barrier within the glomerulus, you can see that there are these three different layers, the endothelium, you know, the basement membrane and podocytes, and they have these different pore structures. And it turns out that if you're about 8 nanometers or so, and now we're getting to about 12 or 15 with our soft nanoparticles versus the majority of this work is actually based on inorganic kind of gold or metallic particles, you can allow them to slip through these different pores and get into the kidney. And so we got to USC, or I guess I got to USC about 6 years ago. And, you know, this is the great thing about collaborations. I knew that this had some sort of potential for kidney, and we were able to then connect with Dr. Ken Howes at USC in the nephrology department to learn about ADPKD and what we can potentially do there. And at that time in 2016, I knew that there, now there's an FDA approved drug, but there wasn't anything available then. And I knew that there was also off-target side effects. And so we developed these micelles, and in particular, we actually added this peptide with 2 lysines and 3 glutamic acids, which is repeated 3 times with the lysine at the end, which is able to target proximal tubule cells, which is kind of, you know, on the earlier stage of PKD. We thought we would place them onto our amplifiers, add some sort of ADPKD drug candidate or drug at the time, and add a fluorescent marker and make these mixed micelles. And we chose this particular peptide because it was found in the literature that they can bind to megalin specifically. This group showed that it binds, and if you knock out megalin, you can see more clearance through the bladder. And they further showed that there is this model drug that typically goes to the liver, but once you put this particular peptide, it can actually redirect it into the kidneys. And so we thought, let's start here. So then we moved on, and we were able to find that it does in fact bind to the human proximal tubule cells with higher efficiency than, let's say, non-targeting nanoparticles. And this is based on, this is all previously published by Jonathan Wang in Nano Research. But the other thing that we noticed was that, you know, there's 2 lysines and 3 glutamic acids. But when we're kind of looking at the GFB a little bit further, we know that it is negatively charged with heparin sulfate, and just kind of going into the literature, we found that more positively charged peptides or molecules could potentially bind or pass into the kidneys a little bit better. And so the other aspect was that this peptide had some promiscuity in the sense that it didn't have to be 3 repeats, it could be 2 and could still enable megalin and kidney uptake. And so we started kind of messing around with this peptide and tried to reverse it with more glutamic acid or more lysines, and started playing around with the repeats. And the idea was, can we make them a little bit more positively charged? And so this is just kind of a snapshot of the full library that we developed. But ultimately, we took the original peptide that had 3 repeats, about 15 nanometers or so, and we went down to 2 repeats and 1 repeat. We also changed the polyethylene glycol linker, which I didn't mention, that was about 2,000 molecular weight, and changed it to 1,000 and 5,000. Then we kind of reversed things and had more lysines, and also did the same kind of math in terms of all the different types of molecular weights and repeats. And we also had some all lysines and all glutamic acid nanoparticles as well. And so you can see the different charges here and the different diameters that we're able to characterize through zeta potential and dynamic light scattering. And just in particular, the one with polyethylene glycol 1000 had the smallest nanoparticle size.
3 minutes.
Okay, sounds good. And so I think I started, oh yeah, right. Okay, so we're just to kind of highlight that we're able to allow for, this is an intravital imaging video that the nanoparticles do go into the glomerulus and then pass into proximal tubules or into the tubules. And we can really skew it using polyethylene glycol 1000 as well as some of these charge-based and skewing them to become more positively charged to kind of get much more into the kidneys compared to the liver, which is where the majority of nanoparticles in the literature oftentimes get delivered to, just based on the reticular endothelial system. Another aspect, though, that I wanted to talk about was that, you know, the majority of particles out there, they're typically IV injected because they've been proposed mostly for cancer applications. And that's a very different scenario compared to the scenario of a genetic inherited disease that lasts for a lifetime. And so we started thinking about, well, first of all, the administration route has to be patient compliant. And the most patient compliant method is oral delivery. But oral delivery also has this type of, you know, low pH and mucosal adhesives or mucosal barriers that don't allow for the most efficient drug delivery into systemic circulation. And so then we started thinking about, well, is there some sort of biomaterial that can potentially load the micelles into? And we landed on chitosan, which is derived from shrimp shells and had been shown in the literature to allow for protection under low pH. So you can see here on the right side that it doesn't degrade or doesn't allow for release versus in more neutral pH, potentially in the intestines or in blood that can allow for more higher release. And so this is just not micelles, but just kind of a model drug rhodamine here. You can see that we just orally deliver free rhodamine that this is the area under the curve in terms of amount present in serum that it quickly drops off after about 48 hours. But when you place it into chitosan, it can kind of have this sustained delivery into systemic circulation. And we found out later that a lot of it was getting kind of, it has mucosal adhesive properties, so it's able to stay within the intestines and allow for a slow release effect. And so I'm gonna kind of skip this slide just so that we can get to kind of the main meat that I wanted to show everybody. This is unpublished data, but just a little bit long-winded, but we took the micelles into that chitosan platform, and that chitosan work is already published in the Journal of Controlled Release last year. And we orally delivered this nanoparticle oral delivery platform. And we find that with increasing, and we place metformin into here, and we find that with increasing sort of engineering, first the metformin on its own, which is hovering around 5%, which is pretty typical in terms of kidney uptake for most drugs. Then you place it into kidney targeting micelles, orally deliver them, and they're hovering around a little shy of 20. Then you place it into the oral delivered chitosan nanocapsule, you can get about 30%. And at the end, when we do this in a long-term PKD1 knockout model, you can see that it does start to make a difference in terms of cystogenesis. This is the micelles within the chitosan versus micelles only, and this is no treatment. And just for time purposes, I'll just show that, you know, this also follows the trend for cystic index and total kidney body weight. All right, so broadly, you know, you can use nanomaterials and biomaterials to pass certain drug delivery barriers that are inherent to most drugs. And right now we have not only proximal tubular targeting, but we have cortical collecting duct targeting micelles that we're developing, which are shown here. We're also using extracellular vesicles and engineering those for PKD. And I didn't really talk about our microRNA and mRNA delivery projects, but hopefully I can share all of these types of efforts that are kind of emerging in our lab. We also have these microneedles that you can use as a skin patch that we've used for nanoparticle delivery as well, and doing some combination therapy. So just want to acknowledge our lab as well as our collaborators at USC and outside. Also, this is a recent picture with kind of the Ride for PKD crew. We have Glenn Frommer here, and this is my lab as well as some of the patients that came to join us on the talk, as well as the funding sources. So thank you again for the invitation, and I'd be happy to take any questions.
Okay, yeah, thank you very much, Dr. Chung. Because we have only 1 minute, let me read the first question. So do you speculate these nanomicelles can pass through the fibrotic regions in the PKD kidneys?
We're not sure. We do have basolateral targeting micelles that we're developing right now. I mean, we've been able to, I think we're doing the immunohistochemistry and getting a little bit more deeper in the histological analysis. But thus far, what I can just say is, you know, the readouts in terms of cystic burden. So that's all I can say at this particular moment. Yeah. Mm-hmm.
Yeah, thank you very much. So I'd like to thank all of the 3 speakers for the wonderful talk. And it's time to move on to the next session 2.